Hello, my name is Catherine Luther, and I'm going to be talking about gene editing. What is gene editing? Gene editing is the insertion, changing, or deletion of specific genes in order to produce more desirable traits. How does gene editing work? There are 4 steps in the gene editing process. Step 1 is design. First, you must choose your enzyme. The enzyme acts as scissors to cut the gene. Cas9 and Cas12 are both the most popular enzymes to use. They are different because of the ways of their cutting. Cas9 cuts more straight and Cas12 cuts more curved. After you choose your enzyme, you must design your guide RNA. Your guide RNA will act as the map that guides the enzyme to the correct part of the DNA so that it knows where to go. Step 2 is delivery. In this part of the process, the Cas enzyme and the guide RNA are delivered to the targeted gene. Step 3, repair. This is when the Cas enzyme will perform what is called a DSB, or double-strand break. DNA is composed of 2 molecule strands, which come together to form what is called a double helix. When the Cas enzyme performs a DSB, a break is formed in both strands of the DNA molecule. This is when a gene can be altered, inserted, or deleted. Step 4 is analyze. It is now time to assess the results of the gene editing. Methods to do this include, but are not limited to, NGS, which stands for Next Generation Sequencing, mismatch cleavage assay, and Sanger sequencing. You may be wondering what these methods mean. Next generation sequencing. This is when DNA or RNA is split into many tiny fragments. Sequencing adapters are then attached to the ends of these fragments. The DNA and RNA information is read from each of these sequencing adapters, and it can read up to millions of them at a time. Next generation sequencing is great because it is very quick and extremely reliable. Mismatch cleavage assay. This is when a target region is amplified using PCR, or polymerase chain reaction. They are heated to separate into different pieces, which can also be called denaturing. They are then allowed to randomly rejoin. The DNA is treated with an enzyme, and then gel electrophoresis is used. This method is not as reliable as NGS, or new generation sequencing. Here is a photo of gel electrophoresis to show you kind of what it might look like. Sanger sequencing. DNA is mixed with a primer. DNA polymerase synthesizes, and then fragments of different lengths are formed. Then, gel electrophoresis is performed to separate these strands into different sizes. Sanger sequencing has a high accuracy and also is a great method to use for gene editing. Here are some real-world examples of how gene editing can be used. Gene editing can be extremely helpful for human health. It can help treat blindness, genetic diseases, and cancer. Gene editing can also be helpful in agriculture. It can make crops more nutritious, have higher yield, and be more resilient. There are many different ways that gene editing can be used and many different methods to do gene editing. It is an up-and-coming science which is very important and can also have pros and cons.